The technique isn't new, the results are. Microscopic genetic material was taken from an adult cell. It was then inserted into an egg whose own DNA had been stripped out. This creates human embryonic stem cells, which are capable of becoming any of the more than 200 types of cells that make up a person. That's important because those cells could be used to treat devastating conditions such as multiple sclerosis, spinal cord injuries, heart disease, or even Parkinson's.
Most of patients don't have the ability to regenerate these tissues, for example, if it's been damaged during disease. Fortunately, now we have a technique where we could take any other cell, in this case, of course, skin cell, and turn them into early embryonic. And now these embryonic cells, they're grown in a petri dish. Now they can produce these heart cells or neurons that this patient needs.
Since the same technique produced Dolly the cloned sheep in 1996, scientists have been trying to create human stem cells. One Korean scientist claimed success in 2005, but this was a lie in one of the most notorious cases of scientific fraud in the last 10 years. The same team behind this latest breakthrough managed to clone monkeys with this technique back in 2007, a significant step. Embryonic stem cell research has repeatedly raised ethical concerns.
What it does is it literally opens the door to in vitro fertilization labs using these principles to make clones for people that might want to have a clone.
The technique used in Oregon is called somatic cell nuclear transfer. What the team in Oregon has done is take an adult skin cell and inserted it into a human egg stripped of its existing DNA. The unfertilized eggs are stimulated by electric pulses, and that starts them dividing. Scientists say the results from Oregon are hugely significant, a major step forward in the field and in the fight against some terrible diseases. Alan Fisher, Al Jazeera.